Good evening, everyone. So this week we'll be discussing basic skin immunology. I have received multiple requests to cover immunology as an offshoot of dermatology, and it is very important area of understanding for the residents because majority of our diseases are either because of immune dysregulation or we utilize immune dysregulation or re-regulation to treat those diseases. Okay, a lot of our diseases rely on using immunosuppressants and immunomodulatory agents. So we should have a basic understanding of immunology as a dermatologist. Okay. Now, immunology is an area of interest of mine, and I thought that I should cover it in much more detail. But since the requests were many, I thought I'll cover a basic aspect, a basic background of immunology in the context of dermatology. Okay. So in this lecture, we'll make a small, we could say a sector of innate immunity, and we'll discuss innate immunity in this video lecture, and then subsequently adaptive immunity and other factors in immunology. And for the members, we will have a longer video which will discuss the role of individual immune cells like mast cells or eosinophils in skin diseases and disorders. Okay, so without much further ado, let's start our basic foray into immunology, and we'll start our discussion on basic skin immunology, part one being innate immunity. Let's start. Skin is known as Grenz organ. Grenz means border. I think it's German. So Grenz means border. Now, skin is responsible for communication with the environment. It communicates with the outside world. And it is also responsible for protection from the environment. So it protects from the environmental insults. Okay. Now, these two functions, communication with the environment and protection from the environment, serves the dual function of skin. And together, this function is termed as skin barrier function. Okay, so skin should act as a perfect barrier to protect us from environmental insults, but also be permeable enough so that communication can happen with the environmental agents. Now, because of this skin barrier function, we have a continuous influx and efflux of immune cells. Immune cells keep on getting inside and exiting the skin milieu, the skin environment. Okay. Now, if we are discussing immunity and skin is the center organ of that immunity in our discussion, the first and important factor is the physical defense and barrier. Skin as a physical layer to protect us from insults. Then we have the innate immunity, which is non-specific. That means this immunity is acts similarly to all the different kind of agents. Then we have adaptive immunity. Adaptive immunity adapts. So it is specific. So it is specific to different allergens. So different allergens will have a different response in adaptive immunity. While in innate immunity, innate means intrinsic. So it is present since birth. And that innate immunity is similar to different kind of agents. They can be decrease or increase in intensity, but the process remains essentially the same. Let's move forward. So before we start our discussion on individual cells, we should know the development of individual cells, and this diagram actually summarizes it very cleanly. Initially, you have the hematopoietic stem cells, and being a stem cell, it has the ability to modify itself into different cell lineages. So hematopoietic stem cells get converted to lymphoid progenitor, which will give rise to lymphocytes. So you have lymphocytes here, lymphocytic lineage. And then you have the myeloid precursor, which will give rise to myelocytes. Okay, so two families, lymphocytic and myelocytic. Clear? Now, lymphoid progenitor will give rise to B cells, T cells, and a specific type of lymphocyte, natural killer cells. Clear? B cell will further give rise to memory B cells and plasma cells. Plasma cells are responsible for formation of antibodies. Clear? T cells will give rise to helper T cells, memory T cells, and cytotoxic T cells, which are usually of two types, CD8 positive and CD4 positive. In fact, helper T cells can be of multiple types, but majorly we learn about CD8 positive and CD4 positive cytotoxic T cells, and they give rise to various kind of autoimmune disorders. Clear? So apart from NK cell, the lymphoid lineage is a part of adaptive immunity. Now, there are exceptions. There are cells in the lymphoid lineage which are part of innate immunity, and there are a certain population of NK cells which are part of adaptive immunity. But more or less, the lymphoid lineage gives rise to adaptive immunity, except NK cells. NK cells are part of innate immunity. Okay, so NK cell is a part of innate immunity. Clear? Let's come to myeloid progenitor. Now, myeloid progenitor will give rise to cells like neutrophils, mast cells, eosinophils, and basophils. The fifth one is monocyte. And monocyte will give rise to two types of cell populations. We have macrophages and we have dendritic cells. Now, dendritic cells are responsible for antigen presentation as cells to lymphocytes. They can also present antigens to mast cells, other dendritic cells, eosinophils, neutrophils. So basically, dendritic cells is the first reporter. Okay. The function of dendritic cell is to recognize the antigen, engulf it, modify it to some extent, and then again put it on the cell membrane so that the other immune cells can get activated and start the immune response. Clear? Macrophages are responsible for eating. They will engulf and destroy the organism. Clear? So we have just summarized the development and little bit of functions of each and every cell. We'll discuss each of these cells separately now. As I've said that in the immunity, we can easily divide it into innate and adaptive. There are further other divisions also, but majorly these two type of divisions are there. So innate system is non-specific. It has lack of memory. For example, if a innate immune cells get sensitized by an antigen, the other population or subsequent population of that cell acts as a new cell, not a sensitized cell. So you have, let's say you have an innate immunity cell and it gets sensitized to an antigen, it will give rise to inflammatory cells, normal inflammatory cells, but it cannot remember the antigen. So every subpopulation will be a new innate cell. Clear? So there's no memory in the cell. The cell doesn't remember that it has already been exposed to an antigen. The process always starts again with each subsequent exposure. Now it is quick. If I don't have to learn new thing, if I don't have to remember and memorize it, I can just just remember it's like in government employee. You are sitting in a government center and all the employee does is just stamp on paper. Okay, he doesn't see what kind of paper it is. The only job is to stamp it. So if I just have to stamp, I don't have to read anything and be specific about it, just stamping, so it will be a quick process. So that's why innate system is quick. It doesn't have to modify itself depending on different antigen. It just have to start the inflammatory process. So innate system does not differentiate between antigen. It is similar response to each and every different kind of antigens. Clear? Now, cells in innate system include granulocytes like neutrophils, eosinophils, mononuclear phagocytes like monocytes, macrophage, dendritic cells, mast cells, and innate lymphoid cells. Innate lymphoid cells are a subpopulation of lymphoid cells which take part in innate immunity instead of adaptive immunity. Clear? Plasmacytoid and inflammatory dendritic cells, keratinocytes which secrete inflammatory cytokines, fibroblasts, sebocytes, endothelial cells, and nerve cells. These are all parts of innate system. Now let's discuss a bit about adaptive system. It has memory. That means the cell remembers the antigen. Okay, if one cell is immunized by any antigen, it remembers and it is ever ready to give a stronger immune response with subsequent exposure. Okay, but it takes time for the cell to train itself. That's why the onset takes about about 1 or 2 weeks. And this system is agent specific. So it's like a government employee again, but this time the employee reads what he is supposed to stamp and then chooses a different stamp for different purpose. So that takes time. You have to read it first, choose a stamp and stamp it. So it is specific to what it's doing. The cells of adaptive system include B cells, sensitized T cells, naive T cells, memory T cells, plasma cells, and dendritic cell populations. Clear? But we'll limit our discussion in this video to innate system. Clear? Let's move forward. So, one by one, we will discuss each and every cell population in the innate system. We'll start first with neutrophils. Now, neutrophils are first responders. They are the first immune cells to reach the site of injury or site of inflammation. Neutrophils are present in circulation and they reach tissues by diapedesis. Now, diapedesis is a process through which circulating immune cells attach themselves to the endothelial cell wall of the blood vessel, squeeze through the wall of the blood vessel, and reach the tissues where they are required. That is known as diapedesis. Now, if there's an injury and I want neutrophils to come there, I'll have to call the neutrophils. And the chemicals that are used to call the neutrophils are known as chemotactic agents. For neutrophils, these chemotactic agents include leukotrienes, complement system proteins, destroyed cells, the parts of the cells which is destroyed and in the circulation in the surrounding region. Now, the destroyed cells can be the cells which are from the pathogen or self, the body cells, all the cells which are destroyed, releases material to call the neutrophils. Now, the function of neutrophils is to rapidly clear the cause of damage. And how does neutrophils clear those damage? Number one is phagocytosis. That means eating and engulfing. If they engulf the pathogen and then using reactive oxygen species, they damage the pathogen. Okay, this damage is known as respiratory burst because it utilizes oxygen. And there are other oxidative enzymes for this respiratory burst, like hydrogen peroxide, hydroxyl ion, and nascent oxygen. Okay, nascent. That means individual oxygen. We know that O2 is the molecular formula for oxygen, but if there's individual oxygen atom, then it is known as nascent oxygen. Clear? The second is neutrophil extracellular traps. So what happens is when a cell is destroyed by infection or inflammation, the DNA molecule will get released in the system. So DNA strands along with antimicrobial peptides, proteins, they come together and they form a structure. And this is a highly disorganized structure. It is just a mixture of all those proteins. And they entrap bacteria, fungus, and viruses. And then this entire structure acts as an antigen to stimulate further inflammation response. And this structure is known as neutrophil extracellular traps. These are traps for the other antigens so that they can collect themselves together, form a structure, and this structure acts as an antigen for further processing the inflammation, for further increasing the level of inflammation. Okay. Sometimes these antigens can mimic autoantigens and can give rise to autoimmunity. So there is an entity known as net-induced autoimmunity because of NAT-associated nuclear antigen. And this has been implicated in disorders like SLE. So if you open Rooks and read about pathomechanism of SLE, you will read about NATs. The third is oxygen-independent mechanism. In this, neutrophils use highly toxic cationic proteins and enzymes like myeloperoxidase or lysozymes to destroy the organism. So it utilizes chemicals to destroy organism, but these chemicals don't require oxygen, so it is oxygen independent. So three mechanisms: phagocytosis, NATs, and oxygen independent mechanisms. Clear? Let's move forward. These are some structures known as ectosomes. Ectosomes are outpouchings of the neutrophil membrane. So these are little vesicles, and what happens is that it will lead to an outpouching, and this will become a small vesicle which the neutrophils will release. Let me change the color. Purple to match the neutrophils. Yeah. So these ectosomes are vesicles or outpouching which are released from the neutrophils. Clear? These are vesicles released from plasma membrane of neutrophils on stimulation by bacteria. So bacteria stimulates and ectosomes are released. There are multiple receptors that can induce the release of ectosomes like membrane surface receptors, CD15, L-selectin, CD66B, 87, myeloperoxidase. All of these are receptors. You don't have to remember, just remember there's an entity known as ectosomes which are released by neutrophils on stimulation by bacteria. And what are the functions of these ectosomes? They can stimulate endothelial cells, platelets, NK cells, and dendritic cells. So to further stimulate other cells. Now remember, in immunity, one process does three things. It stimulates itself, which is positive feedback. It stimulates other processes, which is increasing the intensity of inflammation. And third, it also starts negative feedback so that the process stops eventually. Clear? So always remember, if there is a process, inflammatory process, it will further increase itself. That's number one. It will further decrease itself. That's number two. And it will further increase a secondary inflammatory response. It will further stimulate other arms. So if you have activity in innate immune system, it will stimulate innate system and adaptive system also. If you have problem in adaptive immune system, it will stimulate innate system also. So it is a cross linking. One process is linked to other processes too. Clear? Let's move forward. Now, in the context of dermatology, various disorders require neutrophils. Just remember one thing. Wherever you have neutrophils, you will have pus. Whether sterile or infected, you will have pus. So if you're looking at purulent lesions or pustules or lakes of pus, remember something is happening within neutrophils. You can have other inflammatory cells also causing pus, but majorly it is neutrophils. So disorders like pyoderma gangrenosum, Sweet syndrome, erythema elevatum diutinum, Behcet syndrome, neutrophilic eccrine hidradenitis. Now all of these disorders are termed as neutrophilic dermatosis. In fact, we will find these disorders in the chapter labeled as neutrophilic dermatosis. Clear? Other disorders like SCBD, in which you have pustules, hidradenitis suppurativa, and bacterial infections. Remember, neutrophils are first responders. So that is why and they are part of innate immune system. So that is why disorders involving neutrophils are very quick in onset. They might have a longer duration. For example, a person can have HS for 3 years, but the onset is significantly quick. In 1 or 2 days or 3 days, you start to develop nodules and pustules or sinuses, depending on the intensity, of course. In infections, we can find that the onset is within a week, in few days, because neutrophils are fast acting. They are faster responders. They are first responders. In PG, it increases very rapidly over 1 or 2 days, and with treatment, it can eventually disappear in within a week. Okay, same for sweets or Behcet's. The lesion heals were quite quickly. They come quickly, they go quickly too. That is because of faster acting innate immune system involving neutrophils. Clear? Let's move forward. Now, eosinophils, the red cells. They also do phagocytosis, but it is less effective than neutrophils. Clear? The major function of eosinophils is to protect against extracellular parasites. Now, neutrophils were protecting against intracellular, isn't it? They were engulfing and destroying. But what happens when you cannot engulf? What happens when the organism is so big in size that you cannot engulf? You call eosinophils. Eosinophils have preformed, means they have already formed, preformed cationic proteins, for example, like eosinophilic cationic proteins, and these proteins are toxic to several parasites. That is how eosinophils protect against extracellular parasites, namely nematodes, which are worms. Clear? Now they what they do is eosinophils release IgE antibodies, and these IgE antibodies, let's say you have a worm, these IgE antibodies will coat the worm, will cover the worm. IgE antibodies will cover the worm, and by covering it, they make it more tasty for other immune cells. This is known as opsonization. Now the worm is covered by IgE, and initially, if the worm was not much antigenic, because now it is covered with IgE, it is more antigenic, and other immune cells can come and together, along with eosinophils, they can destroy that extracellular parasite. Clear? Now, after the coating has been done, these IgE antibodies can make the worm or parasite more antigenic. Eosinophils also have receptors for other IgE antibodies, and these IgE antibodies, the receptors namely Fc epsilon R2 or CD23, they also bind to eosinophils. Why? Because remember, eosinophils, even after secreting IgE, has other functions also, namely eosinophilic cationic proteins. Now, how these proteins will be released? They will release by stimulation by IgE. So eosinophils produce IgE for binding to the coating of the parasites. And these Ig, when they bound to other eosinophils, will stimulate eosinophils to release eosinophilic cationic protein. And these cationic proteins are major basic protein, cationic proteins, eosinophil peroxidase, eosinophil-derived neurotoxin. These are all chemicals which the eosinophils will release upon stimulation. And upon our stimulation, it will help to destroy the organisms, which are larger organisms. Clear? Let's move forward. No, majorly eosinophils are implicated in allergic reactions. We know that. Urticaria induce specifically those which are induced by parasitic infections. They're also responsible for recruitment and activation of autoreactive cytotoxic T cells, namely in DRESS or DRESS syndrome. Okay, DRESS. We know that there are eosinophilic vesicobullous lesions or disorders like bullous pemphigoid and bullous IBH, in which you see the cellular population has a rich content of eosinophils. So eosinophilic granules or eosinophilic chemicals can lead to vesicle formation because of inflammation, and we see that in disorders like bullous pemphigoid and bullous IBH. Okay, the inflammation can be severe. There is an entity known as eosinophilic cellulitis, or there are entities like eosinophilic fasciitis. Okay, cellulitis. There's many different inflammatory entities in which eosinophils play a key central role. Okay, and they are mediators of inflammation, but remember, part of innate immunity. Clear? Let's move forward. Now, this diagram is a very good representation of how eosinophils act. Okay, and this is a reference. I put it in reference in description also. You can go and read this article. It's a very beautiful article. Now we know that bone marrow is the source of inflammatory cells or immune cells, or nearly all immune cells. Majorly immune cells come from bone marrow. Now, upon the stimulation of IL-5, remember 5, the fifth letter is E, A, B, C, D, E. The 5, interleukin 5 is for eosinophils. Clear? Now, under the stimulation of interleukin 5, bone marrow releases eosinophils. Under the stimulation, now eosinophils have now gone to the blood vessel in the blood circulation. Now, under the stimulation of CCR3 and very low antigen 4, this eosinophil will attach itself to the cell wall or the blood vessel wall. That is known as adhesion. Now, slowly, the eosinophil will squeeze through the blood vessel. And this is known as transmigration. This entire process is diapedesis. Clear? Now, the eosinophil has reached in the area where you require inflammation. Eotaxins are chemicals which are chemotactic agents for eosinophils. These are signal molecules that call the eosinophils. So under stimulation by eotaxins, eosinophil will release chemicals like TGF-beta. Now TGF-beta will stimulate fibroblast and will increase fibrosis. Increased fibroblasts can further stimulate eosinophils. Remember, every immune process has three things: positive feedback, negative feedback, and stimulation of other responses. Okay, so here what is happening? Eosinophil is increasing the intensity of itself, so positive feedback. It is also increasing fibrosis, the second process. And there will be negative feedback also, which is not shown here. Okay, now eosinophils, when they degranulate, they will stimulate other cells like mast cells. Okay, now mast cells can further stimulate eosinophils using IL-4, TNF-alpha, histamine, leukotrienes, prostaglandins. Again, the positive feedback loop. Now, what exactly does eosinophil does at the site of inflammation? It will release all the granulated proteins, which we have mentioned before. It will also lead to damage by using reactive oxygen species, lipid mediators, and interleukins like 5, 3, and GM-CSF. GM-CSF is responsible for increasing immune cells. Is granulocyte, monocyte, colony stimulating factor. So they will increase immune cells. So increasing the process, the second process. No, because of reactive oxygen species and lipid mediators and the degranulated proteins like eosinophilic cationic protein, major basic protein, okay, eosinophilic peroxidase, all of these chemicals will increase tissue injury and inflammation. So that is what eosinophils are doing. Eosinophils under the stimulation of IL-5 are called from bone marrow. They go through blood vessel and they release their granules, and these granules will cause tissue injury and inflammation. They will also call other immune cells at the site. Clear? Let's move forward. Now, let's discuss the third cell, basophils. Now, basophils are in circulation. That means they are in the blood vessels. They do not reside in a certain tissue. They are just circulating in the blood. They have basophilic granules. So what happened was when basophils were initially discovered, they were discovered by the use of basophilic dyes. So when basophilic dye was used, the granules inside the basophils used to stain with those basophilic dyes, and that's why the name was termed as basophils, cells that likes basophilic dyes. Okay, and eventually it was found that these granules had histamine, so they're histamine-filled cells, which are blue in color, basophilic. Now they are responsible for production of interleukin 4 and 13, and in fact 4 and 13 can itself stimulate basophils. So what we are talking about, interleukin 4 and 13, basophils can secrete them and they can further stimulate them. Again, an example of positive feedback. Now, the granules in basophils have histamine, serotonin, prostaglandins, leukotrienes, platelet activate factor, and these are all chemotactic agents. That means they are responsible for calling the immune cells at the site of injury. That means increasing the second process. Remember, any immune process will do three things: positive feedback, negative feedback, increasing second process. That is what is happening. Basophils, by using histamine, they enhance vascular permeability, bronchoconstriction, inflammatory response. All these processes can be seen because of mediators. By enhancing the vascular permeability, the disorders in which basophils are implicated, you see redness, you see erythema, you see swelling. Okay. The same thing is done by mast cells. Mast cells and basophils are somewhat like brothers, so the function is overlapping. Clear? Let's move forward. Now, we know that basophils secrete 4 and 13, and these are important agents for itching in atopic dermatitis. So basophils are implicated in atopic dermatitis. In allergic response, because it has histamine, immediate type allergic reaction like urticaria and angioedema. Remember, immediate, innate immune system, quick to act, and parasite-induced allergies. Clear? Now, the problem with basophils is they are about 0 to 1% of circulatory immune cells. If you look at any patient's differential leukocyte count, you will mostly see basophils as 0. Okay, so the number is so small. The problem is that we don't really know whether they have any role in human diseases. But the role basically lies in, let's say, in basophilic histamine release assay. Okay, if you study about urticaria, you will come across this term, basophilic histaminic release assay, in which we try to stimulate basophils and see that upon stimulation whether they release histamine or not. And it's an important marker to find out whether the patient will respond better to omalizumab or not. Okay, it's a slightly complex process. Maybe in the future, while I'm discussing omalizumab, maybe I can discuss BHRA, but not now. Just remember, basophils have histamines and prostaglandins and leukotrienes, and they're responsible for immediate type allergic reaction and also as a chemotactic agent to other immune cells. Clear? Let's move forward. Okay. Now this picture is very good. It summarizes all the functions and the physiology of action of basophils. Okay. Now normally what basophil does is upon stimulation by antigens and superantigens. Now these are just protein molecules which are derived from the organisms on the skin. So upon stimulation by basophils, basophils can secrete interleukin 3. And this interleukin 3 further stimulates basophils. Again, example of positive feedback. Now, other cells which can also stimulate basophils are T cells, and they utilize interleukin 3. Mast cells also utilizes interleukin 3 to stimulate basophils. So one cell is stimulating other cell and each other. Okay. There is a slight evidence of using thymic stromal lymphopoietin. This is a chemical, and the thing is that in mouse it was found that it stimulates basophil, but not in humans. So that's why you have a dotted line. The keratinocyte itself can stimulate basophils using IL-33. Now, when the basophil is stimulated, it will further stimulate other immune cells like monocytes, dendritic cells. Okay. Now, what exactly happens is monocyte will transform itself to macrophages. That's what monocyte does. It transforms into macrophages. The dendritic cells will go to the lymph node to stimulate other T cells. Okay, so dendritic cells has gone to the skin draining lymph node and will stimulate naive T cells to form Th2 cells. And that is where basophils are also active. Am I clear about it? So that's what basophil does. Basophil gets stimulated by T cells, mast cells, keratinocytes by the utilization of IL-3, 33, some extent of TSLP. After stimulation, basophils will further stimulate dendritic cells, which will go to lymph node and stimulate T cells. The basophil will also stimulate monocyte to form macrophages. Now, basophil has the potential activity of stimulating all of different immune cells, and that is what it's doing right now. Clear? Let's move forward. Now, the fourth cell population is mast cells. Now, mast cells stain tissues. Remember, basophils were in circulation. Mast cells will stain tissues. Okay, and they have membrane-bound cytoplasmic granules, same as basophilic cells. Okay, mast cells are preformed mediators. That means already formed chemicals like histamine, serotonin, already formed molecules like TNF, interleukin 3, 4, 13. Okay, now the function of mast cells is similar to basophils, but mast cells are greater in number. So you have much more involvement in skin diseases when we are discussing about mast cells. Now, skin-specific or mucosal mast cells have only trypsin, while in connective tissue, the mast cells also have trypsins and chymotrypsins. Why? Because in connective tissue, the function is more towards fibrosis than immune inflammation, while in mucosa, it's more about inflammation. Clear? Now, mucosa comes into contact with the environment, so you need to have a good inflammatory response at the level of skin. While in connective tissue, you need more wound repair, so you have more fibrosis. Clear? Now, mast cells has receptors. It has CD88 receptors, receptor for various cytokines, Fc epsilon R1 alpha. Now, these are all receptors to bind IgE, and after binding IgE, it will release histamine. Okay, we already have a video on pathophysiology of urticaria where I discussed this kind of degranulation with IgE stimulation. You should go and look at it, and you will have better understanding how mast cells releases histamine. Clear? Mast cells also express molecules like CD117, CD26, neuronal cell adhesion molecules, and different molecules. These are also expressed by mast cells. Now, mast cells, we know that under the action of IgE, they can stimulate themselves and release histamine. Other molecules that can stimulate mast cells are PAMs. PAMs are pathogen-associated microbial proteins, and DAMs. DAMs are damage-associated microbial proteins or molecular proteins or molecular patterns. Okay. Now, PAMs are part of the pathogen cell, destroyed cell, and these parts itself can act as antigens. That is why they are known as pathogen-associated microbial patterns. DAMs are part of the self body cell, the cells of the body, which get damaged and these molecules are released. These are known as DAMs. Both of them itself, PAMs and DAMs, can stimulate toll-like receptor 2 and 4, and there are other receptors also. And upon stimulation of toll-like receptor 2 and 4, they can activate mast cells to release these mediators, histamine, serotonin, and various interleukins. Clear? Let's move forward. Now, this slide summarizes the inflammatory and anti-inflammatory action. So I will not go into further detail. The members of the channel I've already discussed with them in detail about the role of mast cells in skin. It is just a summary. So inflammatory processes utilizing IL-6, TNF, alpha, degranulation molecules, degranulation, interleukin 17, histamine, TNF alpha, all of these mediators increases inflammatory processes that we see in infections, allergies, autoimmunity, and increased collagen synthesis, which will further lead to fibrosis. Okay, so that everything has been discussed. While anti-inflammatory processes utilizes chemicals like IL-10, IL-10 and IL-2 and histamine, and these are responsible for decreasing the inflammatory process in allergy and also decreasing the rejection rate of allograft and transplantation. So if you want, I will just remove all the markers and you can take a screenshot of this slide. Also, the reference is below the slide and it is also there in the description of the video. You can go back and read this article. It's a very good article on the role of mast cells in skin disorders. Okay, so this article is here. So I'll just wait 2 seconds when you can take screenshot. Let's move forward. We have discussed neutrophils, basophils, eosinophils, mast cells. Okay. Now we'll discuss a bit about lymphocytes. Now, lymphocytes are of majorly four types. You have NK cells, you have helper innate lymphocytes, T cells, and B cells. We know that T cells and B cells are majorly part of adaptive immunity. Okay. NK cells and helper innate lymphocytes are part of innate immunity. So we'll discuss NK cells and helper innate lymphocytes. So let's discuss first natural killer cells or NK cells. Now, NK cells are cytotoxic. That means they will destroy the cell. They majorly act against virally infected cells or neoplastic cells, and they act via interferon gamma production. Okay, so interferon gamma is the major mediator that are produced by NK cells, and this interferon gamma, if you have gone through our videos on pathophysiology of psoriasis, vitiligo, and alopecia areata, you will find that interferon gamma is a very important inflammatory mediator in responses, in Th2 responses. Okay, now what actually is the function of NK cells? A normal human cell, a normal nucleated cell, has MHC receptors or MHC molecules on its surface. If NK cell comes into contact with a normal cell, which has normal MHC molecule, the NK cell will restrict itself and will not destroy the cell. Remember, these are natural killer cells. The natural tendency of NK cell is to destroy other cells. But if the NK cell gets into contact with a normal cell which has normal MHC, it will not allow itself to destroy the cell. It will stop itself from destroying the cell. But if the human cell, if the body cell is infected by a virus or if it has undergone neoplasia, it will change the MHC structure on its surface. And when NK cell comes into contact with that cell, it recognizes or it fails to recognize normal MHC and will think that something is problematic with this cell. I will have to destroy this cell. And that's what NK cell does. Okay. So MHC molecule on normal cells act as killer inhibitory signals, and there are inhibitory receptors on NK cells. And when these inhibitory receptors gets activated by normal MHC, NK cell doesn't kill normal cells. But if cells are infected or neoplastic cells, the MHC gets altered, it gets different, and NK cell recognizes these different cells and then releases chemicals like perforins and granzymes. Perforins will form holes inside the body cell, and granzyme is an enzyme which goes inside those holes and destroys the cell. Okay, so NK cells are like your security guard. It is going to look at your ID card. If the ID card looks normal, perfect, nothing is going to happen. If the ID card doesn't look normal, it will destroy you. Clear? Now, NK cell expresses Fc receptors which bind IgG, and they can destroy cells which are coated by IgG. This is known as antibody-dependent cellular cytotoxicity. Now, in the first process, we learned that NK cell will utilize MHC pathway to destroy or not destroy a cell. Clear? Other pathways that can use is antibody-dependent cellular cytotoxicity. So either a cell can have normal MHC and that is okay. Okay. If there is any infection in the cell, the MHC gets altered and now it is not okay, the cell will get destroyed. Clear? If you have a normal cell, the MHC is also normal. Sorry, the MHC is also normal, but these cells are coated by IgE antibodies. Let me change the color. IgE antibodies. IgE antibodies. These Ig antibodies will get recognized. I'm so sorry, IgG antibodies. If the cell is coated by IgG antibodies, these antibodies will get recognized by NK cells and the NK cell will then destroy. So NK cell can recognize altered MHC and destroy the cell, or it can recognize IgG coating of the cell and destroy the cell. Clear? I think this is clear. It gives you the basic idea of how NK cell act. Again, NK cell are part of the lymphoid lineage. But in spite of being part of lymphoid lineage, they are part of innate immunity. Let's move forward. Now, NK cell population is divided by the CD56 CD16 expression. You have CD56 bright, CD16 plus minus, or CD56 dim, or CD16 plus. Okay. The first group produce pro-inflammatory cytokines, while the second group has more cytotoxic nature. There are several other subpopulations also, and recent studies have found that NK cells show some specificity. That means some population of NK cell can act as a part of adaptive immunity. So these are just some extra points. Clear? Let's move forward. Now, use of NK cells in skin disorders. Like you have atopic skin lesions in epidermis and dermis. NK cells are found to be there in epidermis and dermis, and they modulate cytokine production of dendritic cells so that dendritic cells can modify a type 2 response. Okay, so type 2 response. So you have atopic dermatitis like inflammation and Th2 responses. Frequency is also reduced, and there's increased apoptosis and infiltration of the NK cells. Now, NK cells are responsible to rapidly destroy and clear any abnormal cells. If NK cell somehow is not able to do that, it will keep on stimulating other immune cells and calling them for help. That I cannot destroy this cell, you also come, together we can destroy the cells. But what happens if the cells which are going to come and help, they are not properly working? You have increased inflammation, and increased inflammation is implicated in disorders like atopic dermatitis and all. Clear? In psoriasis, because of production of interferon gamma and TNF alpha, remember TNF alpha also requires interferon gamma stimulation for it to be released. Go watch our video on psoriasis pathophysiology and you will know. Okay, so interferon gamma produced by NK cells and TNF alpha produced by interferon gamma act on keratinocytes via the receptors for MHC1, HLA-DR, ICAM1, chemokines, and because of that action, there's keratinocyte proliferation. That is what you see in psoriatic scales. Now, keratinocytes can also secrete molecules like camerin and CAMR23 receptors. Camerin is a molecule that keratinocytes express, they release, and CAMR23 receptor is on NK cell. So when camerin attaches itself to CAMR23 receptors, it calls NK cells at the site of inflammation. And when NK cell comes at site of skin inflammation, it will release molecules like interferon gamma and TNF alpha, which will further lead to increased inflammation and psoriasis. Clear? Now we understand how psoriasis is stimulating NK cells to come, and NK cell is stimulating keratinocytes to proliferate and become a scale. It is increasing the intensity of psoriatic inflammation. Clear? I know it's a bit difficult and confusing to understand. It can be a difficult. Immunity is like that only. But I've tried to make it a bit simpler to understand. And if there's any kind of issue, just put a timestamp and just ask whichever process you want to know about again. Now, in allergic contact dermatitis, NK cells are responsible to recruit T cells, produce interferon gamma and TNF alpha after IL-2 stimulation, and it can also lead to apoptosis of keratinocytes. Remember, keratinocytes, the function of NK cell is to destroy other cells, namely, in this case, keratinocytes. Clear? So we have discussed the role of NK cells in skin. Let's move forward. The third part, monocytes. Okay, the lecture is just about to finish. Don't worry, I know it's a very long lecture, but that is how immunity is. Now, monocytes come from myeloid progenitor from bone marrow. We have discussed them in the starting of this video. They circulate in blood and then they reach the tissues. When they reach the tissues, monocytes get transformed into histiocytes. Histio means tissue, histology. So histiocytes, and then you have macrophages. That's how monocytes modify themselves. They are short-lived mononuclear cells with pro-inflammatory and regulatory activities. So monocytes, they modify themselves to form macrophages, but they can also in itself can secrete inflammatory cytokines. There are three types, two major and one intermediate type, depending on the expression of CD14 and 16. They are responsible for formation of inflammatory dendritic cells and macrophages. Remember, monocytes transformed into macrophages and dendritic cells. Just remember that, inflammatory dendritic cells. Clear? Their major function is either modification into macrophages or into dendritic cells. But in itself, monocytes can act as inflammatory cells via phagocytosis and antigen presentation of T cells and cytokine production. So monocytes itself can have inflammatory processes, can itself have an inflammatory role in immunity. It can destroy organisms by phagocytosis. It can present those antigens to T cells and stimulate them. It can also release cytokines to stimulate other immune cells. So monocytes themselves can act as immune cells, and they can also get modified to macrophages and inflammatory dendritic cells and further increase the inflammatory process. Clear? Let's move forward. Now, macrophages. Macrophages are modified monocytes. They as macrophages means large eater. Okay, macro means large, phage means eating, so large eater. So phagocytosis, they engulf the bacteria, they utilize reactive oxygen species, and then they kill the pathogen. So majorly the function of macrophages is to clean the debris. So macrophages have non-specific action. They will come to the site, they will see destroyed cells, they will keep on engulfing and destroy them. Clear? They use mediators like superoxide anions, hydroxyl radicals, hypochlorous acid, nitric oxide, lysozymes, antimicrobial cationic proteins. All these kind of chemicals are responsible to destroy the organism after they have engulfed. Okay. Now, macrophages can also get stimulated by their receptors. Macrophages have receptors. They have receptors for mannose. Mannose is not found on vertebral cells. They are found on bacteria. And that is why macrophages can get activated by proteins like lipoarabinomannan on bacterial cell wall. So mannose is an important sugar that is found in bacterial cell wall. And when bacteria is destroyed, this sugar is released into circulation and they attaches itself to the mannose receptors on macrophages, and macrophages can then get activated by them. Okay. Other receptors include receptors for antibodies and complement proteins. So all macrophage activation can happen via mannose, it can happen via antibodies, it can happen via complement proteins, it can have different sources of activation, and once activated, macrophage will come and keep on eating all the debris and finish it off. Clear? Now macrophages secrete, they release GCSF, which is granulocyte colony stimulating factor. The GCSF is responsible for increasing granulocytes, like all the cells which have granules, neutrophils, eosinophils, mast cells, basophils. Clear? And GM-CSF, that means neutrophils, basophils, eosinophils, mast cells, and monocytes. So what is happening? Positive feedback loop. Macrophage is increasing monocytes. Monocytes will transform into more macrophages. That is what macrophages are doing. So GSCSF and GM-CSF are responsible for growth and release of cells like neutrophils from bone marrow. Clear? Macrophage is responsible for tissue regeneration and repair. So let's take an example. If there's a wall which has been destroyed and you need to construct the wall again, you first need to clear off all the broken bricks and everything. So macrophages are those cleaning crew. They will clean the area, and after cleaning the area, they will start to deposit new bricks. So macrophages are responsible for repair of the organ, tissue regeneration and repair. Macrophages can also act as antigen-presenting cells. They can stimulate T cells, but remember, monocytes, monocytes form macrophages. Monocytes form dendritic cells. The major part of antigen-presenting cell are the dendritic cells. They have the major function. But macrophages can also stimulate T cells. They can also act as antigen-presenting cells, but the capacity is less effective than dendritic cells. So what actually is happening? Monocytes come to the area where you have inflammation. Some monocytes will become macrophages, they will clean the area. Some monocytes will become dendritic cells and they will go back and tell their headquarters that something is happening. Please bring more cells. We need specific T cells. Understand the process? I am a monocyte. I come to the area. I say that some of my group will become macrophages, they will clean the area. Other people of my group will become dendritic cells, and dendritic cells will respond. They will go back to my headquarters and bring more T cells so that we can effectively take care of the injury. That is what the function of monocyte is through macrophages and dendritic cells. Clear? So after that, let's discuss dendritic cells, and I think this is the last immune cells for today. Dendritic cells, the major function is antigen presenting cells, just antigen presenting cells, and they stimulate other immune cells also. Now what actually happens is you have a naive T cell. And this is a specific T cells, or you could say sensitized T cell sensitized. This process is done by dendritic cells. Clear? So we can say the dendritic cell is a bridge between innate immunity and adaptive immunity. This connection is why dendritic cells, of course, different cells of innate immunity secrete cytokines to stimulate adaptive immunity that is happening. But the major junction between innate immunity and adaptive immunity are dendritic cells. So dendritic cells, being a part of innate immunity, will go back and report to their headquarters so that specific action can be taken against problems which are happening and adaptive immunity gets activated. That is why dendritic cells are seen in both arms of immunity, part of innate immunity and adaptive immunity also. We have two types, plasmacytoid dendritic cells and inflammatory dendritic cells. Plasmacytoid dendritic cells are responsible for antiviral defense. That means defense against viral infected cells. They produce chemicals like interferon alpha, interferon beta, which are type 1 interferons, and they also release chemicals like trail ligands or trail TNF related apoptosis inducing ligand, which is a lytic molecule. That means it is responsible for destruction of cells. So, interferon alpha, beta will increase inflammation, while induction of trail will lead to destruction. Clear? These are part of plasmacytoid dendritic cells. While inflammatory dendritic cells produce chemicals like TNF alpha and inducible nitric oxide production, this is responsible for lysis of organisms. Let me write it again. Lysis of organisms. Again, destruction. Inflammatory dendritic cells, inflammation and destruction, both is going to happen. And priming of T cell responses. By priming, we mean sensitization. So this will lead to inflammation, more inflammation. So inflammatory dendritic cells will stimulate more inflammation. It will form more destruction. We have just divided them into two parts. They don't have separate function as such. They do the same thing, but differently through using different processes. So we have inflammatory dendritic cells and plasmacytoid dendritic cells. Clear? Let's move forward. Yeah, so dendrocytes, the processes which comes out of dendritic cells are known as dendrocytes. Dendrocytes come from I think the Latin word dendritic, which means branches or branching like a tree. So if you look at photomicrograph of dendritic cells, you will see these kind of processes. And these processes are dendrocytes, branches. Clear? Let's move forward. Okay, so with that, we have finished a rather long lecture covering about innate immunity, but I think that I tried to make it quite simpler to understand the role of each and individual cells in innate immunity. And if you know what each and every cells is going to do, how they are stimulated, what are chemicals they will release, and how they further the process of inflammation, it will help to develop a good intuitive understanding what all inflammatory process is going on. If you see a lot of pus, think of neutrophils. If you see a lot of itching, think of eosinophils. Of course, there is a lot of overlap between these two clinical markers, but we should have a basic understanding of immunity so that we know the inflammatory process. Clear? These are some reading recommendations. Fitz, Rooks, Bolognia's, and these are some good articles on basophils. Okay, basophils, eosinophils, mast cells, T cells, innate T cells. So if you can, I've copy pasted them in the description. Go to the URL and find the article and read. These are very beautiful articles on each and every aspect. Let's move forward. So again, discussing about Ruby membership, do consider becoming a member of the channel. It helps the channel to grow. It motivates to give more and more uh, newer topics, and it just helps to get this thing going on week after week. And if possible, do consider Sapphire membership. So slowly and gradually over the coming months, we'll shift from Ruby to Sapphire, and you will get much more perks from. In fact, there are about 20-25 Sapphire members as of now, and they have access. They have earlier access to videos. They have access to videos as soon as I upload them. Okay, so you will start to have different perks, better perks in Sapphire membership. So if possible, consider Sapphire membership. Okay, so with that, I will finish this first video series on immunity. We'll come back again next week, and this time we'll tackle adaptive immunity. Now, innate immunity is complicated because it has lot of different players, lot of different cells. Adaptive immunity is complicated because the number of players are small, but the processes are complicated. But still, I'll try and read about it and try to make it much more simpler, much easier to grasp, a bit more intuitive to understand the processes, so that we can develop a good understanding of what exactly is happening with T cells and B cells in the next video lecture. With that, I'll finish. Any comments should be made in below the video along with the timestamp so that I know what you're talking about. Till then, adios and bye bye.